Thanks very much. So while we shift up, I'm going to invite Dr. McConnell to come up and swap computers with us. So Dr. McConnell is an assistant professor of biochemistry and molecular genetics at the University of Virginia School of Medicine. Mike received his PhD from the University of California, San Diego, and performed postdoctoral research at Harvard Medical School in Stanford. His research using stem cells began as Crick Jacobs Jr. Fellow at the Salk Institute, and now he focuses on GNAO1. Mike was actually the first recipient of the Bow Foundation's inaugural grant. We allocated $100,000 to the Bow Foundation to his lab and his research with induced pluripotent stem cells. So he's here today just to give us an update on that research so that we can kind of learn what he's learned and hear directly from him, talk about some of the takeaways and some of the interesting findings, and then where we go from here. So with that, Mike, I think I've given you enough time.
All right, fantastic. Thank you.
Sure.
Hello, it's a pleasure to be here today. It's really inspiring to meet all of you and to talk with you more. I can't express this to Alice and Emily and Duke and Stephen many times over the years, but patient advocacy, I think, is really one of the most important things, particularly in moving these rare diseases forward, so we can actually make an impact from our research. I'll just start with a brief overview of sort of the big picture of what my laboratory works on. So the question I'm particularly interested in starts with genetics. So I'm particularly interested in how genes in your genome unfold and play out to give rise to a brain, and then how that brain actually functions at the end of the day. One tool that I've picked up over my career that Stephen just alluded to are human-induced pluripotent stem cells, and I'll talk more about this in 1 second. But the real advantage of using this technology is that now we can take all of the genes in any individual's genome. They can be an adult, they can be of any age, and we already know if that person has a disease or doesn't have a disease. We call that a phenotype. And so we don't have to know which genes underlie that phenotype, but we know that all of those genes collectively have that, lead to that phenotype. So we can take that DNA, we can make these induced pluripotent stem cells in a dish, and then we can drive those stem cells into neurons. And so we've basically made cells that have all of the same genes as the genes in the patient's brain or in any other tissue in that patient, that let us get very personalized and very specific about what we're studying. And then this is a slide I use many times. So the other aspect of my lab works on some other aspects of how the genetics gets more complicated in some interesting ways. So the essential overview of what we're trying to do is to make iPS cells from individuals in a family, so the patient and both parents. I put an arm here because what we use are skin cells to begin with. So this is a very small biopsy, size of a pencil eraser perhaps. We can take that biopsy and then put it in a dish, grow the iPS cells, coax them to be the iPS cells, and then turn them into neurons. And the point of this is to really get personalized about the medicine. So in the dish, we can look at the neurons, and I'm just sort of cartooning neural activity here, sort of in the context of epilepsy, where on the bottom, the patient will have sort of hyperactive neural activity, reminiscent of epilepsy, whereas the parents don't. And then the goal of the study then would be to take all of the drugs available to us, all of the drugs sort of in Dr. Goodkin or Hoover's toolbox that they might choose, and test them all in the dish instead of in the patient. So instead of going through a trial and error based on their clinical expertise, we can just test everything very quickly, and perhaps even combinations or various things like that, and try to deliver that guidance back to the clinician, who can then take that back to the patient. And of course, at the end of the day, we're all looking to cure and return the patient back to look like the parent. So I'll just show you some background about how this technology's worked. So induced pluripotent stem cell technology was invented in about 2007. It was then awarded the Nobel Prize in 2012. It's really been a revolution in biomedical research. I grew up in science doing mouse research and using mouse models of human disease, And when I started to see this technology come around, I happened to be in a place that was really at the forefront of it. And this is a study that really changed my entire career and pointed me in this direction. So this was a study by Kristen Brennan, who's a friend of mine at the time. A friend of mine sat next to me at the time, and she focused on schizophrenia, so a psychiatric disease. What she did was use 5 patients and 5 controls, and she made the induced pluripotent stem cells from both of those. She turned those induced pluripotent stem cells into neurons, and then she essentially just threw a battery of different assays at these cells to try and figure out what the difference was between the schizophrenic iPS-derived neurons and the control iPS-derived neurons. One of the tests she did was to look at gene expression, which I'm going to point out now because that's what I've done so far with this study. So gene expression, or if I say RNA sequencing or transcriptome analysis, what this refers to is the parts of the genome that are being used in the neurons. So which genes are being turned on in the neurons and how those genes might be turned on differently in a patient relative to a control individual. And what was really exciting about what Kristen found was, I mean, first of all, that she did find a difference. And without diving too deeply into the weeds here, the experiment that she used to find this difference is shown here basically in the amount of red that you see in this image. So the green are neurons in the iPS-derived neurons, and then red are cells that are connected to the green neurons. On the bottom, it's just showing you that there's plenty of neurons in both cases, but the lack of red here is showing you that the green neurons aren't connecting to other neurons in the case of the schizophrenic individuals relative to the control individuals like you would expect. And then what was really important about this is that when she took drugs that were known to help the individuals with schizophrenia, she put those drugs in the dish. She actually reversed that phenotype, as I proposed in the first slide. So what she's showing here on the bottom in this paper was that the control here on the left, I'm sorry, I can't quite see my own slide. So on the top was, so the control shows no effect with the drug. In the patients, you see very little red, so very little connectivity of one cell to another. But then when she treats with the drug, she's reversed that. And so now all of a sudden the connectivity is coming back. It's this drug sensitivity that I think really is encouraging about the iPS technology and this entire approach taking it forward. That's really the bar in my mind of if what we're seeing in a dish is actually something reminiscent and helpful towards what we can do in a patient. Just to show you that this also works in epilepsies, this is a study from Jack Parent's group at Michigan, amongst others, where they took Dravet syndrome individuals, made iPS-derived neurons. Here the flicks up are neural activity, and as you know, epilepsy is hyperactive neural activity. In the controls, they'll see a couple of flicks up, but then in the epilepsy patients, they see this sort of very much what I like to call seizure-like activity in the dish. And then they take this forward and they show that when they see the hyperactivity in the dish, they treat with a drug that can help the patient, and they see that patient and they see that activity go away. So this is in an epilepsy case specifically beyond the schizophrenia. And this technology has been used over the past 5 to 10 years for a host of neurological diseases, everything from bipolar disorder to Alzheimer's disease, Parkinson's disease, and this almost, even to me being optimistic about this, it's been shocking that it just works over and over again. If we find a patient phenotype in the dish, that phenotype in the dish will very often look like something that we know happens in the disease, and then we can treat with drugs that we know help the patient, and that can be reversed in the dish. I think one of the most striking examples recently is in bipolar disorder, where some patients with bipolar disorder respond to lithium, other patients don't respond to lithium. When they made iPS cells from a dozen or so of these individuals, the responders and the non-responders, the phenotype in the dish only worked for the patients that responded to the lithium, didn't work for the other ones. So there's some real accuracy, I think, in this approach. So what we've done, so just to back up and tell you a little bit more about these cells. So induced pluripotent stem cells are very much like human stem cells. So the human stem cell therapies and human stem cell research led to a lot of excitement, basically because from human stem cells, which are originally come from an in vitro fertilization lab or a non-implanted embryo, you can isolate these stem cells, which are a very small population early in development, and they can be turned into any other type of cell in the body. That's the advantage of the embryonic stem cell, right? It's a very small original population that will give rise to the embryo, and in vitro we can actually turn it in specifically to those different cell types. Now, induced pluripotent stem cell technology, we figured out that we can just use 4 specific transcription factors, 4 genes, and we can take those skin cells, which I show a picture of them growing here in a dish, turn them into induced pluripotent stem cells. So that's these big balls of cells that grow in a dish again and look very much like the ball of cells that would be inside the placenta. And then turn these into neurons. And again, as I alluded to before, the real beauty of this is that now we've taken the genome of the patient and we've put that entire genome, all of the genes. So even though we have a candidate gene in GNAO1, some of the differences in the different ways that different patients present might be due to other genes that we don't even know are playing a role in what we would call modifying aspects of the disease. So now by taking the entire genome, we've captured all of that, right? And we've put that in a dish. So now we sort of call this a brain in a dish even. So the study that we've done was to do this in 2 families, one with a movement disorder with this GNAO1 mutation, and one with an epilepsy with this GNAO1 mutation. We've made the iPS cells from both parents and from the patient. The point of this is, again, as I referred to before, we don't know that there aren't other genes hiding in there that are perhaps modifying the effects of GNAO1. So each parent will be contributing 50% of the genes to that patient. And so by using the parents as controls, we're now able to get much more specific and I think much more careful in doing this. And then the goal of the study, of course, was to make the iPS cells, to turn those into what we would call a neural progenitor cell, which is a step before becoming a neuron. Differentiate those. It takes about 8 weeks to get really good neurons, and then we'll do a procedure called RNA-seq. So this is looking at all of the RNA, all of the genes being expressed, all the genes being used in the neurons of the patient versus the parent, and figure out which ones are different, basically. So here I'm cartooning on the bottom. Sort of the hypothesis was that if we look at the genes that are different in the movement disorder relative to his parents and the epilepsy relative to her parents, we might see genes that are unique to the epilepsy or unique to the movement disorder, or some genes that might be common between the two. And so this has been a long process. I've explained to people over the years, so time in a person and time in a dish are the same time. So in order to reprogram these cells, get them to grow up into enough cells, turn into neurons at the end of the day. This has taken the better part of the past year. I think we completed all of the iPS cells last May or June. We had all 6 of these iPS lines made, and then turned them into neurons, and then have done the rest of the analysis. Here's an example of what we get in a dish. I think this might actually be on the Facebook website. So these are the movement disorder patients' neurons in a dish. I'll just point to what we're looking at here. So neurons will often grow in these big balls of cells. I'll just make sure we're all on the same page. Point to a single neuron right here. You can see it as a long process. These neurons are all of these sort of gray dots. There's individual neurons, and they're all sending out processes that connect to the other neurons. So they're forming a circuit in the dish, basically. And then what we've done moving forward is compare these genes. And I think, now that we've done this study pretty carefully, and we're being very conservative in our analysis, but I think that we did find some pretty interesting things. So I'm showing you the movement disorder, what types of genes we found down and what types of genes we found up. So consequences of gene expression can be either that you don't express the gene, or you express it at lower levels, or you express it at higher levels, and go either way. So what we found here, I think interestingly shared between the two, is that we see potassium channel subunits. So these are, when I talk about channels, these are ion channels. These are how the neurons work. This is how they turn into electrical activity. And a lot of the drugs are often targeting different types of channels in very specific ways. I think it's interesting that we see potassium channels down in both cases, And in particularly in epilepsy, we saw many more channels that tended to be down. And likewise, as Rick will talk more about, I think it's also interesting that we've begun to see GABA receptor expression turn up in the epilepsy patient. Perhaps, and so these start to point us towards drugs and categories of drugs that we want to start testing to try and change the activity in the dish more to the activity in the parents. I think one of the surprising things to me are these proteins that might not mean much to you right on the face of them, but protocadherins are a family of proteins that mediate cell-cell connectivity, so how one neuron connects to another neuron, and ultimately the connection of all of these neurons is what forms circuits and what leads to phenotypes and everything else in the brain. And protocadherins are very interesting proteins in this category, and I think it might be the first real clue of how the development is proceeding differently in the movement disorder patients and in the epilepsy patients, in that we actually see more of the protocadherins expressed in the movement disorder and fewer in the patient. And these complement proteins that are down in both cases are proteins that are likewise known to mediate what we call the pruning of synapses. So neurons tend to overconnect first and then cull back and just leave some synapses behind. And so with complement being down in both cases, I think that's quite interesting for a mechanism as to how we're getting to this point at the end of the day. So again, to go back and where we're going moving forward, so the beauty of having done this in the first place with the iPS cells is now we have a limitless permanent resource. So these cells can now be shared with other researchers, and we can keep going back to them and keep doing more studies. So the iPS cells never go away. And what we're doing, taking this forward, as I pointed to RNA-seq in the first pass, now we can start to figure out which cell types are actually doing this. Are they the excitatory neurons, the inhibitory neurons, the glia, the other cells in the brain? We can start to really look more carefully at the activity using calcium imaging, or even using techniques like electrophysiology, where we can record from specific neurons and see really much more carefully at a specific level what's different about the activity. And then, of course, taking this to drugs, which drugs are going to reverse those problems, and then how to take this back to the clinic and to the families. And so with that, I tried to not dive into the weeds too deeply on some of those genes. I'd be glad to answer any questions or discuss any more of those aspects with anybody at any time. But I'll just summarize again by thanking the many folks in my lab, and I especially want to point out Veronica Porterfield, who we have started a stem cell core facility at the University of Virginia now. So this facility actually is the one that received the fibroblast, did all of this work, and has driven it all forward for us to be able to do the analysis. And likewise, Howard Goodkin and Mark Beenhocker are collaborators who I'm constantly talking to and working forward with this approach. And of course, I'm very grateful and very happy to be working with the Bow Foundation, moving all this work forward. So, thank you very much. I'd be glad to take any questions.
We've got time for one or two questions if y'all have any questions for Dr. McConnell.
Yeah, please.
Cord blood? Stem cells? Can you use the stem cells from cord blood, umbilical cord blood, or it needs to be from the parent?
Right. So from cord blood, we can reprogram those to the induced pluripotent stem cells. And we use fibroblasts in this case just because that's kind of the straightforward routine thing that people have done. So it's a very small skin biopsy. But many other sources. So people will reprogram and make iPS cells from blood to do this. It turns out that one actually sheds cells in urine, and you can isolate those cells and use those. That tends to be a slower process because there's not that many cells in urine, so it takes longer. But as far as I know, we've been able to make iPS cells from anything that we've tried to make iPS cells. But I want to just make the distinction. iPS cells and the stem cells in cord blood are different types of cells, right? So while cord blood could be used as a source, that's not really why you bank the cord blood, right? So, yeah, thanks, sure.
So my daughter's had a lot of treatment at Children's Hospital in Boston and MGH, and her geneticist has taken a skin biopsy. I think he's part of something called like the Undiagnosed Disease Network or something like that. Would that kind of information be shared anonymously with you, or would it be helpful to have it? Or I'm not sure where it goes. I know he's doing research on it. and I know he's part of some cohort, but I really don't understand the connection.
So, just from the fact that they took a skin biopsy, and that it's children's in Boston, where I know they do lots of this work, I would suspect that they are making iPS cells, or are at least, excuse me. So from the skin biopsy, you can establish the fibroblasts, the skin cells in culture.
Right, and I know he had me sign disclosure to the community research, but I just don't know where it goes.
So the first thing if it was me, right? Like, so I don't want to speak for them. If it was me, the first thing I would do is to expand those skin that biopsy into fibroblasts, and then I can freeze those and save them, right? So they're probably sitting there as a resource if they haven't already taken them forward to the iPS cells and moving in this sort of a direction. But along those lines, I'll point to a connection that Emily and Stephen had made. A researcher in the Netherlands had already made iPS cells from 4 of his patients with movement disorders. And he and I have communicated, and he's actually sent those 4 lines to my lab. We've started expanding them, and so now we have a cohort of 5 IPS lines from movement disorders, along with the 1 from epilepsy. And at the end of the day, personalized medicine is really what I'm interested in and driving towards, because every patient might be different, but we probably learn the most from what's common amongst all of them. And so having multiple patients with different mutations but the same clinical presentation, I think it could be very useful. Super helpful. And I think that the patient registry might be the place to start doing that. In my mind, that's why we're doing that sort of work. And I think there might be other families in here that I've traded emails with about. I know there's a family in Italy who likewise is connected. Thank you. Hello. So I've connected with their clinician as well, and sort of certainly pooling all of our resources on this. Absolutely.
Thanks.
Yes, one more.
Do you have any cells or stem cells that have both movement and seizure phenotype?
Say that again.
Movements and seizures and stem cells.
Oh, not at this point, as far as I know. We tried to just stay at those 2 extremes, but certainly we can think about the different mutations, and even one of the really interesting things on my mind right now is you've probably heard about CRISPR technology, genome editing in the news. So with these iPS cells, we can do the genome editing in the iPS cells. So what if we take the patient with movement disorder and swap out a different genetic mutation, and do they still have the same phenotype, right? Do they still have the same different changes in the dish? So we can make those changes now with this resource to really start digging into these things.
Thank you. Thanks, Doc.
Sure.
Well, thanks very much.